Welcome to this workshop, to this online workshop this year that is jointly organized by Novo Repair Impact Fund, CARB-X, GARDP, and Wellcome Trust. I'm Ursula Theuretzbacher. I'm now in Vienna and will moderate this workshop. Antibacterial drug discovery and development is very challenging, as we all know, but there are some common mistakes. Therefore, we decided to choose 10 of them in the areas of early preclinical phase, toxicology and CMC, and formulation to present and discuss in this workshop. After each presentation, there will be time for a short discussion. Please use the chat function for questions or comments during or after the presentation. We are happy to have 3 very experienced speakers for this webinar. The first presentation will be given by Dr. Peter Warren. After his academic career at the University of Manchester, Peter was a founder and chief scientific officer of Evotec, focusing on antibacterial PK/PD studies in early drug development. The company was acquired by Evotec, where Peter was senior vice president of anti-infective discovery. He will present now 3 major mistakes in early antibacterial drug R&D. Peter, it's your space now. Peter, I'm sorry to interrupt, but I guess I'm not the only one who doesn't hear you.
Sorry.
Now it's fine. So it was apparently easy to fix. Could you? I think it's okay to just start with the mistake, with the first mistake.
Okay, I'm really sorry about that. I was trying to have good practice and being quiet. Anyway, so the first mistake was with daptomycin, which was the first lipopeptide antibiotic that was discovered by Eli Lilly in the 1980s. There was some fantastic early clinical data in phase 1 and phase 2 trials in the treatment of gram-positive infections. The compound was acquired by Cubist in 1997, and that resulted in an FDA approval in 2003. But if we now go back into history a little bit, a couple of studies were performed by Eli Lilly in the late 80s, and these, I guess, are slightly unusual to the studies we perform now. So in the middle 2 panels, this summarizes a study of the compound in a hamster model of of MRSA infection, and these hamsters had emphysema, so it's a lung infection, and they're able to show that this compound was at least as effective as vancomycin against both Staph aureus and also strep pneumoniae. They then ran a second study looking at 2 strains of Staph aureus in the same model. And in this case, only one of the 2 strains was susceptible. And they put down the lack of efficacy to lack of opsonophagocytic killing of that particular strain. But perhaps that was some small red flag they should have considered. So then the compound was taken over by Cubist in the late 90s, and they ran a whole series of preclinical models, and I'm only summarizing a couple of the very large series of studies they performed. So first of all, they demonstrated they could generate some resistance in vivo, but that this resistance really wasn't relevant, because whilst these had raised MICs, the organisms in vivo still appeared to be susceptible to treatment in rodent models, and that was great news. And then if you look at the bottom 2 panels, in this case they looked at neutropenic thigh burden studies, I guess the bedrock of all the studies that many of us perform in preclinical development, and the compounds had really great activity with a long PAE. They were potently bactericidal, and really this led them to progress this into treating gram-positive infections. So they then went on progressed to some phase 3 studies in community-acquired pneumonia. There were 2 studies. The first was completed in 2001, and the second completed in early 2002. And these were standard randomized double-blind controlled trials. And I'll come back to that in a minute, because these completed in 2002. Before the clinical trials were finally published, a paper came out from Cubist, which indicated that daptomycin had some issues. It had an unusual pattern of killing, in that it was highly effective in pneumonia when that pneumonia was instigated by intravenous infection, in which case the lungs were completely cleared. But in contrast, in relatively basic standard pneumonia models, where the organisms are administered directly into the lung, the compound was ineffective. And there was some great concerns that there was some organ-specific inhibition of the compound. Then if we look at the bottom 2 panels, in the bottom left panel, you can see some in vitro data in which the MIC of daptomycin and ceftriaxone were compared in the presence of surfactant, and even at very, very low concentrations of surfactant, you had orders of magnitude increases in daptomycin. And in the bottom right-hand panel, this was reflected in data from mouse models. BAP is bacterial pneumonia, and these were directly administered to the lung, in which case there was no efficacy at all, in contrast to the rat studies, where there was fantastic efficacy when these were initiated by intravenous dosing. And this result was then reflected in relatively poor outcomes in the clinical trial, which was published quite a few years later, 6 years after the end of the studies, in which they demonstrated that daptomycin had a clinical cure rate of just under 80%, and that compared with ceftriaxone, which was almost 90%. And so this really didn't make the statistical significance that was required for clinical validation. And interestingly, the manuscript, whilst it did mention this, really focused on the difficulties of doing CAP studies, and really this also led to quite a period of time in which CAP studies weren't performed. But how about the lessons? What are the lessons from this? Well, I think that the major lessons are that we really need to look very carefully with the interaction of test articles with host materials, not just protein, which is, I suppose, the standard thing that most of us do, as this can lead to loss or reduced efficacy or total loss of efficacy. If possible, when we test the compounds in vitro, we should try to have a culture medium which replicates the key elements of the host environment, if that's feasible. And I think finally in this example, whilst it's important to minimize the number of animals used in preclinical studies, I think in retrospect, additional rodent studies in bacterial pneumonia might have red flagged the studies and prevented these 2 large clinical studies in CAP. So moving swiftly on to the second lesson, this really looks at frequency resistance. So GSK licensed in a novel boron-containing leucine tRNA synthetase, inhibitor from Anticor, and this was
Yeah, I guess these are the typical challenges of an online conference. And the first year we are doing that, I apologize for the problems, for the technical problems. Our conference organizers are trying to fix this. It seems to be a general connection problem. Usually we read in the newspapers how online webinars and online working, home working is challenged by technical things, and this is real. It started quite well, and now Peter lost the connection, but the organizers are working on it. After this conference, you will have access to the slides, and you can read everything that you may have missed now during this online webinar and the presentation of the slides. So as I see the presentation of Richard Knight, maybe we should switch to the next presentation if Richard is ready. I'm sorry, Richard.
Okay.
We have to be a little bit flexible. But I guess Peter has a problem now, and I hope we can get back to it a little bit later. So apologies to all participants. We are just now jumping to the next presentation. There are enough mistakes that can be done in antibacterial drug R&D. And I would like to introduce now Richard Knight. He's an expert toxicologist with more than 20 years of drug development experience gained in AstraZeneca, where he has worked on projects in multiple therapy areas involving small molecules, biologics, proteins, and oligonucleotides. He's now co-founder and Director at ApconiX. He will present 3 major mistakes in the field of preclinical toxicology. So I would like to ask the conference organizers to start Richard's presentation.
Can you hear me okay, Ursula?
I can hear you. I hope the others can also hear you well.
That's great. Okay, well, thank you, Ursula, and to the AMR conference for inviting me to speak today. ApconiX is a preclinical safety group, and so continuing the mistakes theme, I'm going to talk about 3 mistakes that we see in antibacterial drug discovery that relate to safety. And I'm going to talk about general themes rather than specific examples, but I will use some specific examples to try and illustrate the point. So as we think about safety, it's useful to consider this framework, and that is safety implications from the intended target, safety liabilities that come with the chemical space that we're working in, and then, very importantly, there are the considerations of the indication and the patient setting that pull everything together in order to make the risk benefit. So, mistake 1, and this is something that I would suggest we see quite a lot of, not just in antibacterial drug discovery, but in, I guess, a lot of different indications. And that's thinking about safety really too late in the process. And so we can consider Wile E. Coyote as the poster child or the embodiment of this thinking. Now, I'm sure everyone recognizes that much of toxicology occurs in a well-regulated environment. There are GLP tox studies that support our intended clinical trial program. These are very well described in a raft of guidelines, and then there are a whole variety of national and international guidelines that go along with that describe the safety data that people expect to see in support of a clinical trial program throughout the various clinical development phases, right through from first time in man to marketing approval. But these guidelines are all about characterizing the compound that you've chosen and revealing its properties. But of course, those properties are fixed at the point that that compound has been selected. And these studies are really all about unveiling those properties and learning more about that compound and how we should safely administer that compound to patients. So actually, the time to do something about safety properties and those inherent liabilities that every chemical has is much earlier. Back in discovery, when you still have choice, you have the opportunity to design out those adverse properties. And so this is what we call discovery safety, and it's here that we try to influence compound selection by making safety part of the design considerations of the compound that is chosen to enter the clinic. And it's at that stage that we can bring these 3 things together, as I mentioned earlier. So there are risks related to the target itself, there are risks related to the chemistry, and then we have to pull that all together in terms of the patient-specific considerations before we then can build a bespoke development program. So it's thinking about safety earlier. It's about defining the right questions for your particular program, and they will be different for every program. And then once those questions are defined, it's about choosing if, when, and how to address those potential liabilities. So first of all, I'll just have a few minutes on the drug target. And the aim here is about understanding the target biology in normal physiology. So if we modulate the target in a disease state, what are the consequences for normal tissues? Because that's where adverse events are likely to arise. And there are a variety of tools that we can employ to understand this. Potentially in the literature, we can look at, is our target expressed in the species in which we look? Are there orthologs? What's the expression pattern throughout the body? What are the other consequences that we see from impinging on a certain pathway? Are they genetic phenotypes we can learn from, whether that be knockout animals or human mutations? And does that lead us to suspect specific organ toxicity that we might want to take account of as we move forward? Are there things that we can learn from competitor compounds that have already trodden this path? Are there class effects that we might want to be aware of? Then as we progress through discovery, there are a variety of assays that we can employ, whether this is understanding enzyme selectivity, mammalian versus bacterial. There are various mammalian cytotoxicity assays that we could deploy, as well as particular organ-specific assays. And all these are available for consideration. They're not necessarily ones that we would want to employ in all cases. And then as we get later through development and we start to begin in vivo efficacy studies, or perhaps PK studies, other things that we can learn there about potential safety risks. And it's building an understanding about the space in which we're working to identify potential hazards early, and perhaps that will then influence the suitable selectivity window that we choose. Now, it may be the case that for many antibacterial targets, we have something that's entirely bacterial specific, perhaps targeting the cell wall, for example. And so on-target safety risks for mammalian species might be relatively low or even absent. But for many targets where we're targeting perhaps protein synthesis with a ribosomal target or nucleic acid pathways, topoisomerase, for example, cross-reactivity to mammalian enzymes may be an important consideration. And of course here, I've just picked out fluoroquinolones as an example of, potentially a bacterial target that's effective at topo 2, but can also affect mammalian targets. So it's about understanding that potential, and that may help you then define the requirements for selectivity as you move towards candidate selection. So the next area we might want to think about is understanding the chemical space in which we're working in. And again, this can begin fairly early as an intellectual exercise. And it's about understanding the off-target risks and liabilities that may limit your ability to explore a therapeutic dose in vivo. So we try and identify those hazards early on, and it gives us the opportunity to design them out at an early stage. But of course, first of all, you have to be aware of them. Again, the first step would probably be an in silico approach. There are a variety of SAR databases that are potentially available. There are also a number of in vitro tools that we can deploy. And I think, again, in the antibacterial space, there are some that are more important than others. Cardiac ion channels, for example, is something that we would want to avoid in our chemistry. A lot of people will understand ERG screening, but there are other ion channels such as sodium and calcium channels that we might want to profile in a number of exemplar compounds to understand whether that's a risk inherent in our series. Secondary pharmacology profiling too. So this is an indication of how promiscuous our chemistry is. It might be selective for a bacterial target, for example, but does it also hit a whole variety of other receptors and enzymes and channels and things that we might want to be aware of? Genetic toxicology is always something that's important to understand, whether our compound has the potential to cause a mutagenic change or perhaps DNA damage. Does it have the potential to affect mitochondria? And therefore, does this give us the opportunity to design these liabilities out at an early stage? And then as we progress again towards in vivo studies and efficacy studies, PK studies, if those risks are raised to such a level that it's something that we want to pursue further, those studies then give us an opportunity to look at whether they have in fact translated into an in vitro and in vivo risk. And the other thing to understand, I guess, about antibacterials is that because we generally work at such high plasma levels, the risks that ordinarily might be relatively low for compounds in other therapy areas are elevated somewhat simply because the plasma levels are so high. So we need to consider the top concentrations being tested. For a lot of these examples that I've given, they might tend to have a fixed top concentration, 10 micromolar, for example. But we'd need to consider for our specific case whether that's suitable. You know, there are examples of antibacterials where free plasma levels considerably above 10 micromolar are required. And so we want to take that into account. So just a couple of examples then. This is an example of hERG activity, which is a property that many antibacterials have and often accompanies the more basic chemical series. It carries a risk of a potentially fatal cardiac arrhythmia. This here is a published example by the AstraZeneca group in Boston, looking at aminopyperidines, TOPO2 inhibitors, which had hERG activity, which ordinarily would have been considered quite weak. I think it was 20 to 30 micromolar. Because of the high free levels required in vivo, it did cause an increase in action potential duration in a guinea pig study. So the company put quite a lot of effort into lowering the pKa, which did reduce the hERG activity approximately tenfold from those starting points, while maintaining potency at the primary target, and then that led to successful elimination of that viability in vivo. This is another example, but this time it's the secondary pharmacology profile, which reflects the activity of the molecule at non-intended targets. This one is an antibacterial program, and you can see on the top right-hand side there, compound 2 is quite promiscuous. The solid line is the free concentration required for efficacy. If I can just pick up the pointer. Yeah, so this is the solid line required for efficacy. And as you can see below that, we're already engaging, this is acetylcholinesterase, muscarinic receptors, and some adrenal receptors. And then there are a whole variety of other targets that are engaged before we get to this aspirational 100-fold selectivity over the primary target. So there was quite a lot of work put into building SAR, and before we found compound 12, which had a much improved in vitro selectivity. And as you can see, the solid line here shows a substantial window before we start to engage in these variety of other receptors. And again, the dashed line here is 100-fold over the primary target. So this compound not only had much better selectivity over these secondary pharmacological targets, but then once we moved in vivo, tolerability was similarly improved, allowing us to achieve much higher plasma concentrations. And then the third example here is around genotoxicity. Now, obviously, genotoxicity is an issue. Certain chemical classes are able to interact with DNA, whether that's to cause mutations or to cause direct chromosomal damage. And if that is a property inherent in your series, then you have a very challenging way forward, if at all. And this class of nitroarenes is one such example. It's a class that's known to be mutagenic, and it makes them very unattractive as leads in a variety of drug development settings for that very reason. But they are very highly active as anti-tubercular agents. So in this program, the lead was in fact quite mutagenic, and the team took an iterative approach to building SAR around the active site and aims positivity. And so they were able to design a series of leads with reduced mutagenicity by modulating the electrophilic properties around the key nitro group. And subsequently, that resulted in a series of non-mutagenic leads with a much improved safety profile that retained excellent activity against the tubercular strain. So I hope that first example demonstrates that significant improvements in compound quality can be made in discovery by thinking about the questions that are relevant early on. It's definitely an integrated process that goes hand in hand with all the other disciplines. So we work together clearly with a medicinal chemist in deciding what the appropriate compounds to make, the microbiologist and the DMPK scientist to understand the efficacy and PK relationship. And these activities are all interlinked, and so changing one element often affects the other. So it's about finding the right balance of properties to get the optimal clinical candidate. So why is this important? Why do we need to spend time on this and invest? I think everyone in this audience will realize that we face a huge challenge in our industry, and particularly in antibacterial drug development more than most. I know it takes a huge effort for any program to reach the clinic, and then the reality is that the odds of success still remain pretty dismal. So this is a bioindustry survey from 2006 to 2015, looking across almost 10,000 project transitions, 8,000 projects. With a 19% success rate, but that's still only 1 in 5. And a significant number of those stopped for safety reasons. So we do have to do better. But also I would say it's not just about attrition. It's also about not carrying forward into development those liabilities that we don't need to possess, those that we can identify early and design out. Because carrying them forward into the clinic just gives us an extra challenge that we don't need to have. So it may well limit the dose range, can increase the complexity of your clinical trials, increase patient exclusions and dropouts, increase monitoring, and time and cost. And these are all things that we don't need to have. So time spent in discovery to remove those liabilities is, I would argue, certainly time well spent. So we need to try and stack the deck in our favor, and the time to do that is in discovery when we have those options in chemistry. So mistake number 2, I would say, is not thinking about safety data in the context of the patient. And again, this is something that we frequently see. People think about toxicity as a kind of a siloed activity. You have a GLP study at the end of your discovery process. If you successfully get through that, you can start your clinical trials. I think thinking about your clinical objectives much earlier is definitely the way to go. As I already mentioned, there is work that we can do to identify hazards early, and whether those are relevant to the target or the chemistry, we then need to put those hazards in the context of the patient in order to define the risk benefit. And so this is where alignment with the clinical colleagues really is important. So when we think about the patients, again, it's about defining the risk benefit, and it's different in each case. So it's essential to understand the clinical program, to understand where that preclinical safety data sits. For most antibacterial programs, but not all, we will be going through healthy volunteers in phase 1, and they obviously have a very different risk-benefit setting to patients. We need to think about the route of administration. Is it going IV? Is it hospitalized? Is it oral? Is it in the community? Is it inhaled? And all of these things will impact how we think about the different safety considerations. Along with the duration of treatment. Is it chronic? Is it short term? What about the age of the patient? Are they elderly or are they young? If, for example, you have a fertility issue, it may not be so important if your patient is elderly versus a young child who will then have to live with the consequences of that potential toxicity for the rest of their lives. It's always important to consider the patient context. Just one case example here. So this was a bacterial target relevant to 2 potential clinical opportunities. Again, one was TB, the other was a gram-negative indication, but the series was active at HERG, which, as I mentioned, carries this potentially fatal arrhythmia risk. Now, in gram-negative patients, where the disease indication is particularly severe, significant unmet need, here we have short-term IV dosing in hospitalized patients. Many of those patients will be in the ICU, which gives great monitoring options for watching how that patient responds to the drug. High mortality too, so a particular important risk-benefit consideration. Versus a TB patient in which dosing would be much longer term, at least 6 months. Most of the dosing will be done in the community, where there's much less opportunity for patient monitoring and therefore a very different risk-benefit consideration. So does one compound fit both of those indications, or do you have very different criteria that you need to select for your clinical candidate? And then mistake 3, I would say, is thinking that guidelines are rules. And again, this is something that we often see. There are clearly a raft of guidelines that are relevant to how we support our clinical trial program, and they cover a variety of topics. Things like assessment of carcinogenicity, phototoxicity, genetic tox, different considerations for biological products, for example, and considerations for different patient populations. Is it going into health volunteers? What do you need to have to support women of childbearing potential, for example? What data do you need to support children? And these really reflect the agency's thinking on these topics, but they're not exhaustive. Some of these guidelines are quite old, and they certainly shouldn't be seen as rules. For us, I think they're seen as a starting point, and often it can be a starting point for discussion. So they're not set in stone, and what we find is a lot of regulatory agencies are open to discussion about the data sets that support a clinical trial program. So it needs careful consideration as to how they relate to your particular therapeutic, your patients, and your clinical trial program. And as technology advances, there are many new modalities that are coming into consideration, and not everything fits neatly within a particular box or a specific guideline. And I think we can all recognize times when companies that have followed the guideline rather than follow the science have ended up making huge mistakes. And just to emphasize the point, this is a table from a WHO presentation, I think perhaps based on Ursula's data actually, and it shows many of the different approaches being explored in antibacterial research. And we see a huge amount of repurposing right now, especially in the time of COVID, with drugs moving from one indication to another, novel combinations, changes of routes, duration, dose levels, novel platforms, gene therapies, viral technologies, and other non-traditional approaches. And a lot of very old drugs with preclinical and clinical safety data that we just wouldn't recognize as being up to standard today. All of these are being re-examined in new settings, and so I think it requires us to go back to first principles and ask, what is this data for? And to really start from the beginning, so that the guidelines give us a starting point, but they don't give us the exact roadmap. We need to understand our molecule. What is its target organ toxicity? What's the dose dependency? What's the relationship to exposure? And what's the reversibility? And we can take that information to build a safe clinical trial, regardless of whether it fits neatly into one guidance or another. So it's all about identifying the best possible molecule to give the program the best chance of success, identifying those target organs, thinking about safety as we do every other discipline to get towards the clinic, and then building a bespoke clinical program that gets us down the road further. And I think if we avoid these kind of three mistakes or adopt these three approaches, perhaps, we then get medicines to patients as quickly, as safely as possible, with improved chance of success over the longer term. And then I'll just wrap up by kind of leaving the final word to the coyote. And thank you everyone for listening.
Thank you very much, Richard. You made it through the technical challenges. I would like to ask you one question first, because it's really relevant. Is there a tox approach difference between small molecules and large molecules? Would you do something fundamentally different?
So the principles are exactly the same. The models you choose to address those are different. So for example, the principles are the same. So if we take a drug, whether it's a small molecule or a large molecule, we want to know, where does it go? What does it do when it gets there? Are there adverse consequences? And how do we take those potential risks and build them into our clinical trial monitoring, regardless of whether it's small molecule or large molecule? But there are two fundamentally different approaches. So for a large molecule, like a monoclonal antibody, it has relatively little metabolic safety liability. So those proteins are degraded and excreted. And the majority of the safety risks, for example, with a monoclonal antibody, come from on-target toxicity. So they're very, very selective. So for a monoclonal antibody, it's really all about understanding where the target's expressed, what happens when we engage that target over the short term and then the longer term, and understanding that dynamic. That's quite different from a small molecule, where there are potential for lots of toxicities not related to the primary target, whether that's due to a secondary pharmacology profile, whether it's due to effects of metabolites, for example, and then where do those metabolites go and how do they impact toxicity? So the fundamental difference is around understanding broad potential safety liabilities from a small molecule versus the very selective toxicities from an antibody. But that also means that we have to think really carefully about our choice of species. For an antibody, you should always want to use a tox species that is pharmacologically relevant and expresses the target and engages the target in the same way as the humans. Otherwise, we're not going to generate usable data.
Thank you, Richard. I'm sure there are lots of questions left, because toxicology is maybe really challenging in the early phases. All participants have the chance to ask more questions after the session. So there is this Jitsi coffee room where you can still ask questions to Richard after the session. So thank you very much, Richard. And we are going back to Peter Warren for a second try, and hope that it works this time. He will start with the second common mistake, frequency of resistance. Peter, please.
So hopefully you can hear me. I'm sorry, I was completely unaware that I was that you couldn't catch what I was saying. So hopefully if I see Ursula waving in her little camera, I'll know there's a problem. So if we continue now, I'm just going to go back one slide from the previous presentation, from the previous place I cut off. And so what they found in the clinical study, GSK found in the clinical study, was that there was what they classed as a perfect storm involving very high spontaneous frequency resistance across all the important bacterial species in the range of 1 to 8 times 10 to the minus 7. And not just was there high levels of resistance, high frequency resistance, the resistance generated itself was extremely high level. And in isogenic mutant pairs, they could show over 500-fold increase in MIC. And they an additional problem in UTIs is that you actually have a very high bacterial burden. And also there were some potential issues with the genetic plasticity of the target. And interestingly, they highlighted the bacteriostatic nature of the compound. And it was argued in the manuscript that while some of these factors can be assessed preclinically using in vitro and in vivo models, the clinical relevance of such work is unclear. And I think at the time that was probably right, but perhaps there are some lessons we can learn. But if we go back in time and look at some of the earlier history of this compound when it was still in the hands of Anacor, and there were perhaps some signs that there were issues. In the two right-hand panels are some in vitro frequency resistance data against Pseudomonas and E. coli and Klebsiella, in which the spontaneous frequency resistance was studied at 4 times and 10 times the MIC, and the activity of this compound of the inhibitor was compared to 3 standard of cares compounds: ceftazidime, ciprofloxacin, and tobramycin. In all cases, the frequency of resistance was quite similar at around 10 to the minus 7 for 4-fold the MIC, and about 2 orders of magnitude higher when the inhibitory concentration increased to 10-fold. It was argued that because the Anacor compound was no worse than the other compound, the frequency resistance wouldn't be an issue. But there were some extra nuggets of information in the paper which perhaps could have given some red flags to further studies. In the bottom small panel, I've pulled out a couple of the time kill studies, and you can quite clearly see in the left-hand panel in which the Anacor compound is in blue, and that's compared to the control data in red and ciprofloxacin, which is just about visible in green. And what you can see is the agent at 4 times and 10 times MIC is actually completely static, and it would have been perhaps useful in retrospect to continue the study beyond 24 hours, because actually very large numbers of bacteria remained in contact with the drug. And so what lessons can we learn from this? Well, I think we need to, and I think subsequently, people have actually focused a lot more on preclinical studies of in vitro resistance, and not just looked at target-based resistance, but also looked at the sort of genome-wide sequencing of any bacteria, resistant bacteria that are generated. Because quite often the resistance isn't just in the target, it can be in other parts of the genome. And I think also I learned from this that comparing in vitro frequency resistance rates across classes of drugs and comparing that to a new class of drugs can often lead to misleading conclusions, because whilst meropenem does genuinely have a mutation rate which is very similar, the sort of time kill profile is very different. But finally, and perhaps importantly, I think we should perhaps reconsider the design of in vivo studies, particularly when we see relatively high frequency resistance rates, because quite often the total burden in the very small rodents we use, the mice, is lower than the in vitro frequency resistance rates. So you're never going to be able to identify in vivo preclinical resistance generation. And perhaps we should consider using larger hosts. And to that point, very recently, a porcine model of UTI was published by Nielsen et al., in which they had very high levels of burden, 10 to the 8 per gram, which actually probably equates to about 10 to the 10 bacteria per animal, which may well have identified this problem earlier on. And now going on to the third lesson is, do you have sufficient preclinical data to make a decision to go into the clinic? Eravacycline is, and as an example, eravacycline is a halogenated tetracycline and a new class of tetracycline that's being developed by Tetraphase. Two clinical studies were performed called Ignite 1 and 4, which were in phase 3 for the treatment of intra-abdominal infection. And this led to FDA approval in 2018. But in parallel, a couple of studies called Ignite 2 and 3 were performed in complicated UTI, but these didn't achieve the co-primary endpoints. And as examples of this, in the middle two panels, I've summarized the data from the intra-abdominal infection clinical trial. And in the right-hand panel, you can see, looking at all three endpoints that were used in the study, the microbiological intent to treat, the modified intent to treat, and the clinically evaluable endpoints. In all three cases, eravacycline was at least equivalent to meropenem. Now, if we contrast that to the bottom two panels, which are the data from the complicated UTI study, in which eravacycline was compared to eratapenem, looking at both endpoints in the bottom right-hand panel, both the microbiological intent to treat population and the test to cure. In both cases, the 10% non-inferiority margin wasn't achieved, and therefore the study failed to show clinical efficacy at sufficient levels. But there was quite a reasonable amount of clinical data that was summarized in a manuscript published by Tetraphase, and the bottom three panels show the bottom two left-hand panels are some thigh burden data, and the sort of bottom right-hand panel is some UTI data. And to summarize a very large amount of information in that paper, in septicemia models, eravacycline was really effective against all the species with doses which were pretty low, from less than 1 mg per kg up to 4.4 mg per kg for E. coli. The compound was similarly effective in neutropenic thigh and lung burden studies, where it was equivalent to comparators or better than comparators at doses of between 0.2 and 10 mg per kg. But perhaps a weakness of this manuscript and really the whole of the preclinical package was the very thin data on UTIs. The only model I could find in the public domain was a pyelonephritis model, and this was established following intravenous challenge rather than perhaps the more standard transurethral challenge. And in that study, the drug was highly effective and showed almost 5 logs of reduction in burden in the kidney. But I would argue that this data is much more matched to septicemia data than UTI data. And so if we now just look at some of the parameters and perhaps look at how this could be avoided, in the left-hand panel, I've covered very briefly the PK of ribavacycline. The compound has great linear PK. It's got 28% bioavailability, which is pretty reasonable. The driver of efficacy is AUC over MIC, and the magnitude, depending on the publication you read, but it's somewhere between 10 and 30. The compound hasn't got that high protein binding. It's around 80 to 90%, which is very similar to tigecycline. The renal excretion is a little bit lower, only about 50% of tigecycline at 16%. But interestingly, the urine concentrations in the phase 1 study in healthy males was pretty reasonable, showing levels really over the first 24 hours post-dose of around 5 mg per ml. Total drug. So that's about 0.5 mg per ml a free drug. But a couple of years after the clinical trial, David Nicolai's lab generated some really nice data showing that a mouse dose of 2.5 mg per kg IV was pretty, in terms of AUC, was equivalent to the human level. So that number is very important. 2.5 mg per kg is equivalent to the human levels after a 200 mg per patient dose. I then went back to look at if there was any more data, and I could only find really one more decent publication that was worth referencing, and this was a poster at the 2010 ICAC, which I've summarized in the 4 right-hand panels. The top 2 panels are models of pneumonia, neutropenic pneumonia in mice, with Streptococcus pneumoniae in the left-hand panel and MRSA in the right-hand panel. And in both cases, it was possible to achieve efficacy at doses which weren't very far away from the human dose. And these strains had pretty low MICs of 0.016 and 0.063. The bottom 2 panels are 2 studies of pyelonephritis following intravenous challenge, the bottom left-hand panel being E. coli and the bottom right-hand panel Klebsiella pneumoniae. With E. coli, with a reasonably low MIC of 0.13, efficacy, modest efficacy was achieved at the doses which are easily achievable in humans. But interestingly, in the right-hand panel with the Klebsiella strain, it took 20 mg per kg, which is 8-fold the human dose to get good levels of efficacy. So what lesson can we learn from this? Well, I think we've got a huge amount of experience in the community translating mouse thigh and lung efficacy data to clinical studies, and we can do that with a lot of confidence. This has been done for the last 30 years. But the volume of data on translating mouse UTI studies is much thinner, and we all see now, as particularly if you look at sort of CARB-X funding, many programs are focusing on CUTI of multi-drug resistant gram-negatives, and I think we haven't got a lot of experience in this, so a great deal of care should be exercised in extrapolating these results, and really to get over that, I think we need a much larger package of UTI data, and we all accept that these experiments are complicated, they're expensive to run, but I think the volume of data we generate should be very similar to that generated in thigh or lung models. That's to say, we probably need 3 or 4 species and 4 or 5 examples of each of those species, and probably 8 or 10 dose fractionation studies to give us confidence that we can move forward into UTI, because this is really a very new area, and is particularly complicated because whilst E. coli UTI models are reasonably straightforward and lead to very robust models, UTI models with Klebsiella, Proteus, and Pseudomonas are rather more challenging. But that said, I think we do need to focus on it. And with that, that's my final lesson I've got time to present. Thanks very much.
Thank you very much, Peter. It went great, technically speaking, so no problems. We could hear you very well. so far I have not received any questions, so there is still room for 1 or 2 questions if you want to send them. If not, you can always send them also afterwards to the organizers, and we will find somehow a way of addressing them, because I think these are so fundamental issues, and especially if you think about the funding situation, that's not really very favorable right now, and it's very tempting to do some shortcuts in especially the early phases. But you may be lucky and get away with it, or you may fail later on. So it's really very important to know where you can afford a rather thin package, or where you need to invest in more preclinical studies. So in interest of time, I would suggest, so thank you very much, Peter, for this presentation. In the interest of time, I would suggest to move to the next and the last presentation that will be given by Aleksander Danilovski. He's Chief Scientific Officer at Xelia Pharmaceuticals. That's a specialty pharmaceutical company focused entirely on anti-infective treatments with production sites in Europe, and that's very special. He led the development of numerous active pharmaceutical ingredients and the development of finished dosage forms. He will present 3 major mistakes when developing formulations and CMC strategies. So, Aleksander, are you here?
Can you hear me?
I can hear you, but I cannot see you.
Okay. Here we go.
Okay, thank you. It works fine. Please start your presentation.
Okay, so thank you so much, Ursula, and for the introduction. And I will try to very briefly go through the mistakes in the antibacterial, but in general, but I will focus for this talk on the antibacterial development, which are related to CMC parts and also to the formulation part. So it's a bit switching gears, you know, from the predecessors, which or very nicely summarized a lot of things that can go wrong on, let's call it, biological front. But there are also some things which are quite also challenging that can delay or even kill any development, and that's as well to the antibacterial part. So I will very briefly, for the sake of time, go through the setting the scene part and then show you the 3 case studies. So I think we all know that, and I really don't want to focus on this slide in details, but I would like to dedicate this talk to the part of all these challenges we have in the anti-infective arena, to the operation part, but on the R&D side, because it also later on relates to the potential manufacturing challenges. So, very well-known slide, but pretty much the discovery funnel of any program, not just antibacterial, from discovery, preclinical, clinical, into commercialization. And I borrowed this wagon and horses from a colleague from the Novo family, Alex, because it very nicely describes, you know, the whole value chain that we have in the antibacterial, wagons being, let's call it, the R&D engines, horses, the traditional commercialization fronts, but it all sits on something which I propose to call the wheels, which is the underlying foundation of the whole program, which has to do with the drug substance or API, drug product or the finished dose, and also formulation related to it. And as you will see on the case studies, there are examples where these aspects can kill a program or even can be a reason that we have a false positive or false negative indication in the antibacterial development. So again, don't want to bore you with details, but it's a bit a busy slide. But basically, companies do invest a lot of money, and this is just example of two companies: Cubis, that was already mentioned by Peter before in a more trial setting example, and Paratech, where you can clearly see that it's a lot of money, and that's the main takeaway, that companies put on a table to bring product to the market. It's around $500 million that covers the whole R&D piece, regulatory and launch. And in that part, there is a sizable portion of the CMC spend, of course, related to the clinical trial material, but also when it comes to launch, to preparing for the launch, making launch quantities. So it is a sizable spend, and it's worth to be, let's say, considerate of some of the challenges this can bring. So going to the case studies, and basically I will have a privilege to show you, in a way, a personal case studies that I was involved with. So one was a case study which originates way back in time. So we are talking over 20 years ago. It's related to a big pharma setting. So it was old in terms of, because it's a development before year 2000. It was a new chemical entity program for a broad spectrum, which was having a known mechanism of action. So it was derivatization of a known class, and the compound was in a lead optimization step. The compound as such met all initially defined target product profile targets. So it was very well characterized, or at least we thought so. It was fitting the specification. Also, it was exceeding all in vitro targets that we set against all primary and secondary panels. Also had very promising in vivo animal efficacy in 2 animal models. Because of that package, we as a company decided back then to advance this compound to the next stage, where we were planning to do some more detailed PK and safety profiling, as was alluded earlier. Safety profiling with just a bit of a detour of a 20 years ago approach, and we needed more material. And what we then do, we just went back to the chemists and said, please make us more, I think was close to 50 grams of material. And newly made material was again passing what we had as a specification, but it failed all in vitro assays. And it failed dramatically. So as is written here, the MIC values went from below 0.5 against various bacteria, which was the initial values that we have from the compound, and the new values were all above 16. So a sizable difference. It's not one dilution. It's significantly more than 1 dilutions. Then, as usual, we said, okay, maybe it's an error. We confirmed it 3 times with 3 different teams, both on the chemistry and biology front. And unfortunately, it didn't confirm the first, but the last result, compound was inactive. And then it caused a lot of grievances in the company, a lot of tough meetings. And then we said, fine, let's do the final test. And we appointed a third party, both on the chemistry and on the testing side, on the biology side. So we synthesized the compound by third party. We did the MIC, we didn't do the animal part. And unfortunately, the third party again confirmed the inactive results, meaning that the compound absolutely, the real numbers were more than 16, not below 0.5, and basically compound was inactive. This was after some, I think, over 5 years of various chemistry activities on the bench. And because we wanted really to dig deep what went wrong as a lessons learned, we performed a very detailed and lengthy root cause analysis. And it was almost, when what you watch on movies, the CSI. We went through, human factors to chemicals, a lot of stuff. We went that deep that we also picked up the solvents. So what we have found out, and just to cut the long story short, that the solvent was the culprit. And it's on a way that this initial compound was made with a solvent that basically the bottle that was used for making that solvent was consumed. So there was no more solvent of that bottle. Yes, the solvent was ordered from the same supplier, but it was a different batch. And those different batches of solvent simply gave the compound that was inactive. Luckily, there were some traces somewhere deep down of that original bottle of solvent, and there was an impurity in that solvent, basically that was killing the all bugs, not the compound. And we absolutely didn't have that in the specification. So yes, our material was past that, and that impurity, which was then also we profiled, it was also toxic, so that's why we couldn't go further with it. But basically, as simple as little solvent, or sorry, an impurity in a solvent that you use in organic chemistry, left, right, and center, was the culprit for the activity. And once repeated, the material was inactive. Obviously, the project was stopped and all activities ceased. But this was a huge learning for us back then, some 20 years ago. Case study number 2 is related to a smaller company, a small biotech company. Sorry and apologies, it's not before 99, it's before 2019. It's a typo. So it's a small biotech startup which was founded in early 2010s. It was, as usually, founded and the seed capital was raised on the initial discovery from some university laboratories. It was a novel spectrum, novel compound, novel mechanism of action, and it was in a mix in between lead ID and optimization step. Again, very similarly like the first story, the compound initially made did meet all the set TPP targets. It also was very extensively profiled on the mode of action. And as on the previous case, we needed more material to progress further. And because this was now a company, not a university outfit, and the company was more, let's say, centered around biology part, we immediately went and hired the third party to make these needed quantities of API for further profiling. Unfortunately, third party CMO could not even reproduce the university notebooks. So basically, the procedure for making the compound didn't yield material, or it yielded some mixtures. It was a purity challenge and a lot of various synthetic challenges. All how the problems never go in one, they go in pairs or in triplicates. The person that made the initial compound at the university labs, who made the initial also notes in the lab notebooks, left the university. The quality of documentation was extremely poor on that part of the CMC front. So then CMO basically went to, in a way, to reinvent and modify the recipe. They managed, after a lot of efforts, to make some material, and this material was, again, like in the first story, completely inactive. This was repeated by another CMO, who again used now the procedure from this first CMO, made the compound was tested, and again inactive. Unfortunately, in this case, because it's a biotech setup, and when these results were there, and when one saw that basically it's unreproducible, basically company was in a way liquidated. And due to also a lot of human resources, it was impossible to do a detailed and lengthy root cause analysis in this case. If one were to summarize these two case studies, basically, and what could be the key learnings and takeaways, is that quality is extremely important here. And here I don't mean quality that now should be like the real commercial stage CGMP type of thing that will, in a way, create a lot of obstacles and delay progress, but there needs to be a consideration around quality aspects. And GMP, and this was, I remember one of my colleagues who in a way, didn't like GMP at all, called it a great mountain of paper. But in many ways, it is something that could be a gatekeeper if done appropriately, because you really want to have procedures that are reproducible. You really want to have a documentation in place that if you progress to third parties, you are able to make the material. You want to control as much as you can, again, appropriately, your starting material. You want to have appropriate recipes, analytical methods that will be able to in a way detect potential impurities or byproducts that maybe could be the culprits of activity. So it should be done in appropriate way, but absolutely it should be more way of thinking, because there is more examples, and we don't have time than these two, where GMP aspects, or I wouldn't say GMP, CMC aspects, or reproducibility, quality of material, analytical aspects, killed great programs. Moving to the third case study is related to the formulation, and here I will just briefly share some of the survey that was done by Informa. And here I would like, it's a courtesy of Leucocare, one of our partners we work with from Germany. Basically what was done, there were around 150 people that were interviewed and that were participated in a questionnaire, who then answered the various types of formulation related questions in drug development. What was very interesting, as you can see, 60% of respondents said that they have experienced either a project failure or a delay because of a formulation challenge. And when one digs deeper into what those delays and failures were, so this 60%, if they are then more broken down, it was 38% of those 60 had that they basically encounter less than 12 months delay. 52% had more than 12 months delay, whereas in 10% of respondents, the formulation aspect were the reasons why the basically activities were completely stopped due to formulation challenges. Digging further into it, and then they were asked in which stage they do apply formulation. Expectedly, it was said, as you can see with the red colors, it was during phase 2 and before phase 2B, or during phase 2B before phase 3. And this was the majority of respondents. And then what was basically the importance of those formulation aspects, as you can see here: competitive advantage, reduced time to market, and I would say as well reduced probability of failure in the drug development process, were rated as the most relevant elements for the formulation importance. Also, respondents were asked, and I don't know if you can see it, but the colors are from deep blue, it's more than 6 million, and then it goes to 1 to 5 million, 500,000 to 1 million, and then we have 100 to 500,000, 50 to 100K, and below 50K. How much they invested into formulation development. So this is the current stage, and then what they are planning to invest in the next 5 years. As you can see, it's almost unanimously, all of them plan to almost double. If you look, all the spend above million, or even triplicate the amount of money spent for the formulation development. What is here in contradiction to the previous slide, where they said that they are doing it at a quite late stage. And going to the here key findings and learnings. So clearly, many basic NC discoveries, formulation is done quite late. It is, as said here, phase 2, 3, before phase 3 for sure. Some do it before phase 2. But then if you pair it with the amount of failures that you have, with the amount of delays you have due to formulation challenges, one would say that probably it's done too late. And early stage formulation could be extremely beneficial, Not just that it will help a successful commercialization later on, but it will also help enable to solve some of the activity problems, because we shouldn't forget that formulation is not just excipients mixed with API. Those excipients have an extremely important pharmacological efforts, and that's why if one basically starts with the formulation trials early enough, you are also increasing changes of success as well. You know, you are adding beneficial IP, time to market. You can also do a lot of in the ease of use. You can plan your life cycle management accordingly, because I was also part of my career in the generic side as well, and I can tell you that formulation was the favorite area where generics were digging holes into any new products. And therefore, if one doesn't do that right, you are also leaving yourself exposed that after all these great $500 million spent to bring the product to the market, you are risking that some agile and a formulation expert generic company can potentially find an alternative way and launch a product. So the key findings and learning from that would be to start it as early as possible, again appropriately, but definitely not wait after phase 2A and before phase 3, and do it right the first time. And with that, because of time, I would like to wrap up this, Ursula. So over to you.
Thank you very much, Sasha. I think that was a great overview of a field where a lot of things can go wrong. And I guess that many of us are thinking mainly about activity and safety, and that this very important field may be neglected, but it can have really bad consequences. So I would like to ask our participants to write your questions. So far, I have not seen any. I hope that I'm not missing them. Yeah, I don't think there are any. You may also have the opportunity to ask some of questions in the coffee room, in the online coffee room. You got all this information from the conference organizer. But thank you very much, Sasha. That was very helpful. And I don't think that we have a shortage of mistakes. There are a lot of them. So if you count it with me, then we had 9 mistakes so far. And because the number of 10 is nicer than 9, I try to find to decide which one would be relevant. And according to my experience over the last years, I noticed that the field of potency is also prone to some potential mistakes. And it all starts with MIC testing, and especially for new classes and totally new compounds, it's sometimes really difficult to know what MICs mean. I have seen a lot of projects with rather high MICs. It could be also paired with high protein binding and not really existing PK data. And concluding from this information that a drug has a high potency may be a little bit early, especially if there are no PK or safety characteristics known at this time. So if, in general, assuming that the MIC testing was fine, then if you have high MICs, then you need to balance that. You need really great PK data, PK and safety data, because otherwise you need a very high dose to make up for maybe not the most optimal PK characteristics. And that's probably more difficult than when you are starting with lower MICs. Just to illustrate what that could mean, here you have MIC distributions from old drugs, and you can see that the MIC values could be very low in case of penicillin or ciprofloxacin and also meropenem, but quite high for piperacillin. So MIC distributions as such don't mean very much because you don't have the PK data. So for piperacillin, because it's very safe and you are using very high doses, you could make up for not the best potency. But I would really recommend to not assume that not the best potency is a good starting point, because it means potential problems with PK or safety. So with this, in interest of time, and also because you have afterwards the opportunity to ask questions in the private chat room, I would like to end this topic now, but would also like to ask you to send me or the conference organizers some ideas of potential common mistakes that you think are very important and define success or failure later on. So if you want us to consider more of these mistakes or failure analysis for future conferences, then please contact me or the conference organizers, and we will try to plan for that. So to conclude this session, and I hope you could follow it with not too many technical issues, I would like to thank all the participants for their patience, and also our speakers and the technical support. And I wish you all a fruitful and inspiring AMR conference, and good next days. Bye.